Okay, chapter 10. Now, the next two chapters are going to talk about something, are going to, I know, I get it, are going to talk about cell reproduction. We're going to talk about, in chapter 10, a particular type of cell reproduction, and then in chapter 11, we're going to talk about another particular type of cell reproduction. Chapter 10 is going to involve the type of cellular reproduction that we call mitosis. And mitosis is very simple. Mitosis is no more different than taking a, did I turn the recording on? Yes. It's no more different than taking a piece of paper or, say, your class notes to the copier and running an exact copy of it. That's what we're making. When we undergo cell reproduction in mitosis, we make clones. The cells are identical to one another, okay? Almost identical. I'll talk about that in just a second, okay? The next chapter we're going to get into, chapter 11, which is the other type of cellular reproduction, which is meiosis, is how our bodies produce sex cells, sperm or egg. If you think about the number of chromosomes, it makes sense that meiosis would have to occur. How many chromosomes do humans have? Do y'all know that? How many chromosomes do we have, typically? Yeah, we have 46. That's right. 23 pairs. Very good. Yes. Now, Tulsi hit that nail on the head with the 23 pairs. We really like to refer to them as 23 pairs because you remember you got a pair from your father in the sperm. We call those your paternal chromosomes, and the ones that came from your mom would be your maternal chromosomes. Let me look. No, nobody's got their mic on. Hi, Seth. How are you? Good. Alrighty. Maybe it was me sitting too far away. I don't know. Let's see. All right. So in this chapter, we're going to go from cells. We were talking about human cells with 46 chromosomes, and they're going to create cells with 46 chromosomes. In the next chapter, chapter 11, you can already sort of get an idea what's going to happen, right? If the sex cells, if your sperm and your egg, if male and female, obviously, if they both had 46 chromosomes, how many chromosomes would the offspring have? And I'm hitting you with some pretty heavy-duty math early in the morning, right? So 46 chromosomes in the sperm, 46 chromosomes, yeah, they'd be 92, and that's not good, right? We talked about balance all semester long, and when it comes to balancing in terms of chromosomes, having the right number of chromosomes is essential. It's very important. So what's going to happen in the next chapter, in chapter 11, is we're going to divide the number of chromosomes in half. Your sperm and your egg only have 23 chromosomes. We're also going to make each one of those variable. Now, that's in chapter 11, so that's where we're going to. But in chapter 10, I want you to understand that chapter 10 is simple cell reproduction. All we are doing is making a copy of a cell, an identical, quote-unquote, identical copy. And I'll get in why I'm putting that in quotes in just a second. First of all, let's talk a little bit about why we want to have this process of cellular reproduction. Well, for one thing, if you're a single-celled organism, that's the only way you reproduce. So maybe that's an amoeba up there. It's kind of a weird-looking amoeba. But if you were a single-celled protozoan, the only way to, and if you're a single-celled prokaryote, the only way for you to perpetuate the species or to have offspring would be to divide, right? And so that would be reproduction. Tissue renewal. You know, if you get hurt and some of your tissue's been damaged and it needs to be replaced, then you would generate cells to replace that tissue. Cells also get old, don't they? Right? They're entropy. Entropy affects any system, and cells are a system. So when they get old, maybe they're going to be replaced as well. Growth and development. You know, after the sperm and the egg come together, then it's time to take those 46 chromosomes, that single-celled, and divide and divide and divide and divide until we have a multicellular offspring. Up in the right is actually kind of a cool picture. These are two animal cells dividing. And we'll talk a little more specifically about what's going on in the middle here. But basically, when we get toward the end and those cells are ready to actually split into two, it's almost like you take the purse string on a purse, you know, that you pull in tight, or a cinch. And there's a series of proteins that are wrapped around the middle of those cells up there. And they're basically pinching, getting tighter and tighter and tighter until you pinch that cell in half. excuse me. This thing over on the right-hand side is a group of cells called a morula. When organisms develop, as the cells are dividing, that's supposed to be an O, there comes a point early on in development in which it's a mass of cells, a morula. I like that word, morula. I think that's neat. In Latin, it means a group of grapes. So that's kind of what that looks like, is a morula. So if you ever need to ask for a group of grapes in Latin, now you know how to say it, okay? All right, so those are the reasons behind why we do that. Now, let's take a look at the organisms that reproduce by this process of mitosis, but let's specifically look at the prokaryotic organisms first. All right, so we'll look at the prokaryotic, which is actually very simple, and then we'll look at the eukaryotic. Prokaryotic organisms divide by a process that's called binary fission. I want to name my next fission. Oh, there you go. That'd be a good name. So prokaryotic cells like this E. coli cell, we're all very familiar with E. coli. E. coli is a bacterial cell, divides by binary fission. Binary is like binary code in computers is zeros and ones, and fission is to split. So 1 makes 2, right? And then down below, if we were to continue on with this graph, each of those would divide. My pen works, and then it just stops. I don't know what that's all about. And those are going to divide into 2, and then those are going to divide into 2. And they can do this pretty rapidly. That's the one thing about prokaryotic cell reproduction. If they find the right environment, many times that's, say, the intestines of your body, that's a very good environment for them to reproduce. And they'll start one. I looked for the number the other day, and I couldn't find it, but I believe there's an E. coli in one day can produce something in the vicinity of 2 million offspring. I might be exaggerating that a bit. It might be 200,000 offspring, but whatever it is, it's quite a bit. Yeah, we don't do chapter 9, Jordan. We didn't. Yeah, we did skip it. All right, so let's run through this process. Up at the top, we've got an E. coli cell, and if you remember from our discussions about the difference between prokaryotes and eukaryotes, we know that prokaryotic cells have 1 chromosome, right? We got 46. Other species have other numbers. Prokaryotes have 1 chromosome, and that chromosome is in a single loop. So if we took all that purple spaghetti stuff that's wrapped up in that cell up here, and we unwound it, untangled it, it would be 1 single loop of DNA, 1 single loop of chromosomes. All right, now it makes intuitive sense that if you're going to divide the cells, that the cells are going to each have to have a copy of the chromosome, right? This original cell only has 1 copy. So in order to make sure that each resultant cell has the chromosomes, and you got to have chromosomes. Chromosomes are the instructions for the cell. A cell without chromosomes cannot survive. So what you're going to have to do first is make a copy of that chromosome. You can see that's what's happening in the next picture. It says 2 copies of the origin. What they mean by that, let me get that pen out of here before I grab the wrong one. Mother Nature is extremely efficient. So if this was the DNA, it's double. Right, 2 strands to it. The way the copy is made is you start at the point of origin, and the copying process goes in both directions until it comes around and meets at the other end. So it's not like you make copy by going around the circle all the way. No, they start from the middle and work around the other ends. Very efficient. So as we're moving down, like you see in picture number 2 there, 1 copy of the origin is now at each end of the cell. So now we've got 2 pieces of DNA. We've met at that other side of that circle, and we've now got 2 chunks of DNA. So now we're ready to do the other part. Now we're ready to fission the cell or split the cell. So the cell splits in half, and now we have 2 daughter cells, and each of those has a copy of the chromosome. Pretty simple, right? Not much to it. The only thing you really got to think about, and again, it really sort of makes common sense, you're going to copy the chromosome first, and then the cell's going to copy. So you're going to split the chromosome, not really split, but copy into 2 parts, and then you're going to split the cell. Makes absolutely no sense to do it the other way around, does it? Now, just as a little bit of a side, when we talk about in Chapter 11, and I mentioned this a few minutes ago, the purpose behind Chapter 11, there's a few purposes, but one of them is to make variable cells. The sperm and the egg, every one that you create is a little bit different than every other one. And if you go on and you take Bio 2, I talk a lot about variability, but variability is essential for living organisms. if you look at this, and this process here, and the process of mitosis, actually produces clones of cells. So they're ostensibly genetically identical to one another. People often wonder, well, I don't know how many people wonder, but some people wonder, well, then how does E. coli end up being variable? Variability is important for living organisms, because if everybody's exactly the same, like the poor cheetahs. Cheetahs in Africa went through what we call a population bottleneck about 10,000 years ago, where their numbers dwindled down to just a few. Same thing with elephant seals. Elephant seals used to number somewhere in the vicinity of 20,000, and when they were hunted by humans, the lowest point they ever got was down to 20 individuals. And so the rest of the elephant seals out on the California coast are descended from the original 20. There's not a lot of difference between them. Lack of variability among cheetahs, lack of variability among elephant seals makes conservation biologists a little nervous, because if everything's identical, everybody's identical, then if a disease comes along, like we talked about the plague, remember we talked about how the plague killed a third of Europe, but 2/3 stayed alive. And why did they stay alive? Presumably they were exposed to it. Some of them would have been naturally immune to that disease. But if everybody's exactly the same, then nobody's immune and the whole thing can go extinct. So how do the prokaryotes get around that? Right? They don't do meiosis. They don't make those variable cells. As I showed you here, they make quote unquote identical cells. Well, the process of DNA replication, starting up here, and we'll talk more about DNA replication in some subsequent chapters, but the process of DNA replication is not perfect every single time. So when we go through and we split the chromosomes in half, and of course, you know, remember talking back in that chapter, it's always an adenine that binds to a thymine and a guanine that binds to a cytosine. I love my dog too. So if you split the chromosome in half, maybe that's one side of it. This would have been the other side. And then all you do when you're making copies is you just add the other base to it. See, now we got two copies of the same DNA molecule. All right, now that's supposed to be identical copy. Well, nothing is perfect in the world. Even the process of DNA replication, making exact copies, sometimes does not make an exact copy. There'll be a mutation somewhere. One of the letters will be changed. One of the letters will be moved. Now, if this was my Bio 2 class, right after that, I would then say, well, what do you think happens to most of those organisms that get this random genetic mutation? And hopefully they would all answer the thing that I've tried to drill in their head. They would probably die. Yeah, when you mess with the genetic information, most of the time that is detrimental to the critter. Most of the time, okay? Now, how do E. coli get around that? Well, I said most of the time it's detrimental, but not all the time is it detrimental. So E. coli, if they're producing, what did I say, 200,000, or maybe it's 2 million, whatever the number is, a whole boatload of E. coli, if in those organisms that can produce lots and lots and lots and lots of offspring, they can actually deal with these random mutations. Even though most of them are going to be detrimental, there might be that one out of 200,000 or one out of 2 million that has a mutation that actually gives it an advantage. So that's how we, among other ways, viruses help prokaryotes in terms of variability too, but that's one of the ways we get variability. The DNA replication process isn't always exactly perfect. Now, there are enzymes that actually move down the DNA molecule, kind of like those guys that down the train tracks with the hand car, and they move down the DNA molecule and they try to repair these mistakes, but sometimes the mistakes are more than the ability of the cell to repair them. So we get a change. Now, in prokaryotes, that's going to be a much more pronounced change, because in prokaryotes, all of their DNA is red. All of their, not RED, but all of their DNA is used by the cell. In eukaryotes, like you and I, it's only about somewhere in the vicinity of about 5% of our DNA is actually read and turned into proteins. The other 95%, and years ago I used to call it junk DNA. Everybody used to call it junk DNA. It's not junk DNA anymore. A good chunk, well, some chunks of that 95% we're finding out are regulatory genes and things like that. But suffice it to say, regardless, in eukaryotic cells, there's a lot of DNA that's just there. Doesn't do anything for you. It doesn't code into a protein. It's just there. Why does it continue to be copied over and over again? Well, to be honest with you, the best way to answer that is probably refer you to a book known as The Selfish Gene by a guy named Richard Dawkins. It's a short little book. Yep, it's there. Richard Dawkins talks about the selfish gene. You see, DNA is a really unique molecule. It can make copies of itself. That's not something that molecules typically do, right? Glass of water, 1 million water molecules. You don't expect to come back in an hour and have 2 million. You're probably going to have less because some evaporated, right? But this DNA molecule does have the ability to make copies of itself. And so when DNA replicates, all of the molecule replicates. So the molecule DNA, I always tell my students, your DNA cares nothing about you. You are just a carrier for that DNA molecule. You are just a vehicle, if you will, by which to make lots of copies of that DNA molecule. So those things that quote-unquote junk DNA, or that 95% that has no function, or whatever portion of that 95%, most of your DNA, regardless of the percentage, is not DNA that's read. It's not DNA that's used. So that's why that stuff is still around. Now, if a mutation occurs during DNA replication to us eukaryotes, and it happens to that part of that, let's just keep calling it junk DNA, right? If it happens to the junk DNA, no problem, right? It doesn't make any difference. That DNA is not read, so if an A got switched for a T, or a G for an A, or whatever mistake occurred, so what? Nothing happens, no harm, no foul. It's only going to be a situation when it has an effect when it occurs on that 5% that's actually read, okay? Now, prokaryotic cells are different. All of their chromosome is read. All of their genes and all their chromosomes are read. So any mutation is going to have an effect on that bacterial cell, on that prokaryotic cell. And as I said, remember, most of the time, those mutations are going to be bad. They're going to affect the cell negatively. But if you produce a lot of them, right, it's a numbers game. If I know that 1 out of 1 million mutations will be good, right, beneficial, okay, so 1 out of 1 million, so that means 999,999 are bad, 1 out of 1 million. If I make 2 million, I got 2 good cells. Right? And I've got 2 cells with successful mutations. Mutations are like rolling the dice, genetic mutations are, folks. It's random. You might get something good, you're probably going to get something bad. But the prokaryotes, with their rapid reproduction rate, can take advantage of the fact that most of the time it's going to be bad, because every once in a while there's a good one in there, right? It's kind of like the lotto. What are your odds of winning the lottery? Something like, well, you choose 6 numbers, right? And each of those 6 numbers, you can choose 1 to 47. Is that correct? I haven't played the lotto in years. So there's actually a way to figure out your odds of winning the lottery, if it is 47 numbers, is 1 divided by 47 raised to the, oh, wait a minute. Yeah. Raised to the 6th power. There's no calculator. If you're going to play the lotto this weekend, let's see what your odds of winning. Now, if you buy 1 ticket, right, this is your odds of winning 1 ticket. 47. Oh, shoot, this calculator doesn't even do higher powers. So we got 47. Oh, I can't see the doggone thing. Somebody do 47 to the 6th power for me, please. This doggone calculator's got the little keys in blue, and I can barely read that. It's some big number. So it's 47 times 47 times 47, 6 times. I don't want to do that. Big ass number is what it is. 1.07 times 10 to the 10. So it's 369. Good Lord. Anyway, you got a better chance of being hit by lightning while you're being chased by a polar bear, right? Did I? Sorry. All right, so again, though, I always tell folks, you want to double your chances of winning a lottery? Sure, buy 2 tickets. It does. You get 1 over 47 to the 6th with 1. You get 2 over 47 to the 6th with 2. You still got a lot of numbers, combinations that won't match your numbers. But every once in a while, somebody does win the lottery, don't they? All right, so think about that prokaryotic cell. Most of the time, they're not going to win the lottery. But every once in a while, somebody gets the right genetic combination that gives that organism an advantage. Is that the number 10,779,215,329? So that's your chances of winning the lottery. If it's 47, because there's 6 numbers you pick, and I think you have between 1 and 47. So that's your chances of hitting the lotto. Good gosh. What the heck? Like my dad says, you can't win if you don't play. Yeah, so... And I don't play very often, but every once in a while from the 7-Eleven, I might grab one. All right, so that's prokaryotic cells. Let's turn our attention now to eukaryotic cells. It becomes a little bit more complex in eukaryotic cells, if for no other reason than we got lots of chromosomes, like in the case of humans, 46. Watermelons have somewhere in the vicinity of 1,000 chromosomes, I believe. So having the number of chromosomes really does not have anything to do with how advanced you think you are. Tobacco plants, I think, have 78 chromosomes, which is more than our 46. All right, so let's orient ourselves here in this picture and talk about some of these terms, because I'm going to use these terms a lot as we go through this discussion. They can become kind of confusing. All right, these red things here are chromosomes, and what they've done is they've been condensed. And we'll talk in just a second about when this happens. But the reason why you condense chromosomes is because it's easier to move them. And that's what you're going to be doing. You're going to be moving them around the cell. You're essentially going to be doing the thing we used to do when we played kickball. You guys over here on one team, you guys are over here on the other team. We're kind of going to do the same thing with the chromosomes. So the chromosomes are going to condense, but before they condense, they're going to make a copy. So you can see in the picture there's 2 chromosomes, right? All right, now there's a little bit of confusion, so you got to pay attention to what I'm going to say. There is things called sister chromatids, and then if you look down below, there's something called a metaphase chromosome. All right, now what's the difference between a chromatid and a chromosome? Nothing. There's no difference between them. It's where they are, all right? You're only a chromatid when you are attached to your copy, like you see in the picture there. And you're attached to your copy because there are proteins that hold you together. They're called aptly cohesion proteins. You can see them right there. See, that's the little yellow in between. So whenever this process of mitosis occurs and these chromosomes are condensing and making copies, of course, you're going to have to make a copy just like in the prokaryotic cell. When that chromosome is attached to its copy, we rightfully call it a sister chromatid. And there are 2 sister chromatids. But you'll notice in the block down below, they call it 1 chromosome. If that's a little confusing, think about a chromosome. No, not quite. A chromosome contains a specific piece of information, and both of these are copies. So think about that term metaphase chromosome referring to the information that's on those sister chromatids. Let me go back to Seth's question. So the chromatids are the individuals, and the chromosomes are chromatids bound together. Chromosomes are individuals, right? You have 46 chromosomes, and when they make a copy and they're held together, then they're called a chromatid. Does that make sense, Seth? So chromosomes are only called chromatids when they're in this arrangement right here. Most of the time when the cell's not undergoing division, each of these are individuals, the paternal and the maternal chromosomes, and they're doing their thing that they need to do to keep the cell going, right? But when they're getting ready to divide and they're bound to their copy, they're going to be called sister chromatids. You can see a picture up here too. This is a, just going to get rid of the pen because it's just frustrating me. There we go. There's 2 sister chromatids. All right, centromere. The centromere is not a structure. It's a region. It's a narrowing of the chromosomes, right? You can see right here in the picture up above, it looks like the chromosomes begin to narrow there. Now, centromeres don't have to be in the middle, like you see in the picture here. They can be up towards the top of the 2 chromatids. They can be near the bottom. Wherever it is, it's the narrowing part. And at that narrowing part is where the cohesion protein that's holding them together. Now, the little green things that you see attached, those things that are called kinetochores, those are proteins that are attached to the chromatid. And they, in turn, are attached to things called kinetochore microtubules. Now, you might not have seen the word kinetochore before, but I know you saw the word microtubules. Anybody remember discussing microtubules? When did we discuss microtubules before? Yeah, cell structure, right? We talked about the components of the cytoskeleton. You remember the microtubules and the actin filaments and all that sort of stuff and the intermediate filaments. Those all things had a variety of jobs. They gave the cell structure. They helped move organelles around. And one of the functions, if you remember when we were discussing it, was it talked about moving chromosomes. All right. Now, so these kinetochore microtubules are attached to the kinetochore, and the kinetochore is attached to the sister chromatid. Now, those kinetochore microtubules, when they attach, are actually pulling. So the pair of sister chromatids is already under stress in this particular picture. Those microtubules are wanting to pull those two sister chromatids apart, but they can't because of those proteins, right? Those cohesion proteins do exactly what it says they do. They cohesivate, not a word. They stick those two chromatids together. All right, but it is under tension. And I kind of give you a little foreshadowing because a little bit later on, we're going to get rid of those proteins, right? And then those chromatids are going to separate. This is a cool picture up here. I love the way they do the fluoroscopy and microscope. They get different things to glow in different colors, like the green right there, those are the kinetochores. This little red area, that's the centromere region. And then the blue are the chromosomes around it. Very neat. Down here below, and this will become a little more applicable when we get into meiosis, so I asked how many chromosomes we had earlier, and somebody said 23 pairs. Yeah, the paternal and the maternal. So, for example, in this picture, the blue ones would be the paternal and the red ones would be the maternal chromosomes. And that's an actual picture of chromosomes that have been dyed or probably artificially colored afterwards. 950x, so these things are pretty doggone small. In a chapter. Thank you, Jordan. All right, let's talk a little bit about what's going on here. How do we get these condensed chromosomes? So how do we get from a DNA molecule, which we looked in the nucleus of a cell, DNA molecule is like a really thin thread. There's all these DNA molecules scattering throughout the nucleus of the cell. They're all very thin. And we're going to move them, so we're going to condense them. Condensing them makes it more efficient to move. Think about moving yourself. If you're moving from one apartment to the other and you had to move the dishes, there are two ways really to do it. One is completely inefficient and one makes sense. You could move one dish at a time, right? You could take one dish and move it to the new apartment and go back and get the other one. Or you could, quote unquote, condense the dishes. Could you put them in a box? So you put all the dishes in a box and carry the box over there. So it's more efficient to condense the chromosomes. But let's talk a little bit about how this condensation structure arises. So take a look at the next slide. Start with something we're familiar with. There's the DNA. There you go. Same idea. Yep, grab all those bags at once and take them instead of keep going back and forth. So there's our DNA molecule, and DNA, of course, you know, is an important molecule, and it's a relatively fragile molecule. So we're going to protect it. It's like if you were shipping crystal glasses to somebody on the other side of the United States. You wanted to make sure they didn't break. You might put those little white peanuts, the styrofoam peanuts that protect it. Well, DNA has its own little form of styrofoam peanuts. They're obviously not made of styrofoam, but it's a protein called a histone. And if you look inside, this is the histone protein. Outside is the DNA. So the DNA wraps around the histone. Now, when the DNA is wrapped around the histone, it's unreadable. And a little bit later on in the semester, we'll start talking about how DNA is read and used by your body. And what it does is it unwraps from the histone, it's read, and then it's wrapped back around the histone again. All right, now a group of these histones with the DNA wrapped around them is called nucleosomes, and the nucleosomes coil into a fiber like you see there. So the little yellow part in the picture would be the histones, and then the blue or bluish gray colors would be the DNA wrapped around. And they're going to wrap themselves into a fiber. And those fibers are going to wrap up and eventually make these little rosettes. This red thing in the middle here is a scaffolding protein. The little rosettes of DNA are going to anchor around that scaffolding protein. Now, if you have a bunch of those rosettes all crammed very close to one another, you're looking at a cross-section to a piece of that DNA. So the scaffolding protein runs right down the middle, and all those little rosette loops are crammed next to one another. So you don't see between the little rosettes, but that's what you would buy there. There'd be all these little rosettes right next to one another. Be there. Creatures of habit, right? Keep grabbing for my pen. Rosettes upon rosettes upon rosettes upon rosettes with a scaffolding protein. There's a lot of them, so it looks like one solid thing. All right. So now we got our terminology. Any questions out? We all understand chromatids, chromosomes, kinetochores, all that kind of stuff. We're good with that? Because I'm going to be using those words a lot as we go through these different steps. Very good. Thank you, Courtney. All right, let's go simple. Let's start with a cell with one chromosome. All right, eukaryotic cells, you're not going to have something like that. And this is not a prokaryotic cell, but we're just going to focus on one chromosome. So it's one chromosome, which is a double helix of the DNA molecule you see there. The first thing that's going to happen is we're going to duplicate it. Okay, remember like we talked about in prokaryotes, you got to copy the DNA before the cell splits. Makes no sense to do it the other way around. So we're going to duplicate that chromosome, and then after that chromosome's been duplicated, those sister chromatids are going to separate so that each resultant cell down there at the bottom has its own chromosome. The term we use to refer to this process, chromosome duplication, that is known as karyokinesis. Kary, K-A-R-Y, nucleus, prokaryote, eukaryote. Eukaryotes have a nucleus. Prokaryotes don't have a nucleus. Kinesis, potential energy, kinetic energy, energy of motion. Karyokinesis literally means nuclear movement. Now, it's not the nucleus that's moving, right? It's the nuclear material. It's the DNA that's moving. As a matter of fact, you look in this picture, the nucleus is nowhere to be found, and the nucleus does disappear for a period of time. But first, we're going to get karyokinesis, right? And so in karyokinesis, we're going to duplicate the chromosomes, and then we're going to separate the cell. And when you separate the cell, that's called cytokinesis. So karyokinesis is followed by cytokinesis, not the other way around. We're good? Now, your cells don't spend all their time dividing. If you think about yourself and your life, right, you don't spend all your time engaged in reproduction. I mean, I guess if you added it up at the end of your life, you might find out that percentage-wise, you spend a very small percent of your life engaged in any sort of action that would involve reproduction. In spite of the fact that, where did I read? I can't remember where, that 17-year-old boys, psychologists tell us, think about sex once every 20 seconds. It's amazing. Or once every 20 seconds. That's right. 17-year-old boys do. In spite of that, as you get older and the rest of us, you do got other things that you need to do. The cell needs to do cellular work or the chromosomes need to do cellular work. The chromosomes are the instructions on how to run the cell. So we have something called a cell cycle. And the stage I'm talking about in which the cell is just kind of doing its regular duties is known as interphase. Interphase is made up of three substages called G1, S, and G2. Now, it's kind of an interesting story. The G1 and the G2, the G came from, for a number of years before our microscopes became as good as they are now, we didn't know what was happening during this time, so we called it gap. So that's where the G comes from, gap 1 and gap 2. Now we know quite a bit more of what's going on. So let's go through the interphase phase first. Now, interphase, you notice up here is the mitosis. This is when the dividing. There's cytokinesis. But as you see, that's a small part of that cycle. So let's go through each one of these steps. During the G1 phase of interphase, the cell grows. The cell's just divided, right? And so it's going to grow. It's not going to grow too much, right? It's going to grow a little bit, doesn't want to get too big, wants to stay small, wants to maximize that surface area to volume ratio like we've talked about, so it's not going to get too big. The cell is also going to differentiate. And what we mean by differentiate is the cell is going to become what type of cell it needs to be. So if it's a heart cell, it's going to differentiate into a heart cell. And if it's a liver cell, it's going to differentiate into a liver cell. And what I mean by differentiation is all the cells of your body have all the same genes. The cells in your heart have liver genes. The cells in your liver have heart genes. It just depends on which genes get turned on. In the heart, in those cells, the heart genes get turned on. In the liver cells, the liver genes get turned on. That's what I mean by differentiation. We're going to turn on and turn off those genes for whatever the intended purpose of that cell is going to be. All right, now these stages are dynamic, so even though it looks like there's a line between them like that, it isn't a set line. A lot of these processes are still going on when the next process occurs. So the cell is still growing. As a matter of fact, you notice the cell grows throughout all of interphase. Even in G2, it's still growing. But in S, during the S phase, it says DNA synthesis. During the S phase, the cell is getting ready to divide. Now, if you think about the case of human beings, or for God's sakes, the poor watermelon, you got 46 chromosomes or 1,000 chromosomes to go through, and you got to make copies of them as accurate as you possibly can. So even though you're making copies for this eventuality up here, you're going to start doing that back in the S phase. You're going to start making copies of that DNA. So the DNA is going to be copying, it's going to start condensing, it's going to start doing all that stuff that we talked about when we looked at that picture of the sister chromatids. So DNA synthesis is still going on when we move into G2. DNA synthesis doesn't actually end until we actually get into mitosis. During G2, the cell is going to continue to grow, but also during this time, the cell is going to it's going to get ready to divide. It's going to begin to accumulate energy. It's going to begin to accumulate certain proteins that it needs to actually do the dividing process, okay? But we're still not dividing yet. We're preparing for it, but we're still not ready to divide. So if we looked at a cell in G2, like the one on the left-hand side, and if we looked in the nucleus, we see the chromosomes are duplicated, but in this case, they're not condensed yet. So they still look like a big string of little threads going everywhere. You look in the picture next to it, which is the next phase after interphase, prophase, you see the chromosomes are now condensed. When we get in the lab and we look at the onion cells under the microscope, you'll be able to see the chromosomes. And you'll be able to see them, well, one, because they're dyed or stained, and another one because they've condensed, they become thicker. All right, now, I got to talk a little bit about a second between the difference between animals and plant cells. The stuff I've written in red on this slide, if you look down below, only involves animal cells. Animal cells also have centrosomes. Remember, a centrosome was a region when I talked about cellular structure. Not really a structure. It's a region. And in the centrosome, in that region, there's a pair of these little things called centrioles. And they arrange themselves in pairs at right angles to one another. Okay? Now, in animal cells, the centrosomes and the centriole pairs are going to split. So now we're going to have two centrosomes and two pairs of centrioles. And around the centrosomes, these little things that are sticking out, these are actually microtubules, by the way. That looks like a star. Those are called asters. All right, now, make sure you pay attention. Asters, centrosomes, centriole pairs, only in animal cells. Plant cells do not have them. They perform a function. When we get a little bit later on, we'll talk about that. But the plant cells can perform the same function, but without these structures. So you're not going to find asters or centrosomes and centriole pairs in plant cells, only in animal cells. Okay? All right, so we're going to move out of interphase and move into prophase. When we get into prophase, this condensation that was starting during the S phase of interphase is finished. So now we've got condensed, duplicated chromosomes. We have two sister chromatids of one chromosome. So let's do a little exercise. How many chromosomes are in the nucleus of that cell? How many chromosomes? Chromosomes. Not a human cell, Seth. Courtney, chromosomes. Chromosomes. How many chromosomes do we have? Courtney, yeah. And how many chromatids do we have? There you go. There you go, right? 6 chromosomes, 12 chromatids. Human beings then, with Seth said we had 46 chromosomes. How many chromatids are we going to have in prophase? There you go. Thank you, Tulsi. All right. Now, another thing that happened during prophase is as these, in animal cells, as these centrosomes with those central pairs pull apart, these things called mitotic spindles begin to form. Now, there's another word for mitotic spindles, and let me introduce that to you now, so in case you hear it, you don't get confused. They're also called spindle fibers. All right, and spindle fibers are made up part of those cytoskeleton structures. So as those centrosomes and central pairs and asters begin to pull apart to opposite sides of the cell, these mitotic spindles or these spindle fibers begin to form. Now, plant cells do the same thing. Plant cells produce these spindle fibers, but they don't do it between asters or centrosomes and central pairs, okay? And again, I'll come back to why there is a difference between plant and animal cells as it involves that. But a plant cell would look identical to this except for these pieces up here. There and there. And it probably wouldn't be round, it'd be more square shaped. Under a microscope, this is what prophase looks like. You can see the chromosomes are condensing. Again, that's really the best time to see them under a microscope when they're condensing because they're thicker enough. There's the nucleolus, by the way. This is all the nucleus. If you look over to the left, that cell looks like it's just a little bit further behind. That condensation hasn't come along quite as far. All right, now, as I said at the beginning, these stages are dynamic. It's not like one stage ends, boom, and the next stage begins. These stages kind of blend into one another. When I was in advanced genetics in college, we actually learned, you guys are going to know 5 stages. You got interphase and prophase. We've got 3 more to go. But we learned something like 16 stages. It just kind of depends on what you're looking at and you know what's going on. This next stage is kind of one of those in-between stages. This is what we call pro-metaphase. The next stage is metaphase. We just came from prophase. So pro-metaphase is kind of in between. And what's happening in pro-metaphase is if it's an animal cell, those central pairs and those centrosomes have made it to opposite ends, those spindle fibers have formed, and the nuclear envelope is beginning to disintegrate. See, we need to do karyokinesis. We need to move those chromosomes around. And you can't move them around when they're confined in a membrane. So we're going to get rid of that membrane. It's going to come back. Don't worry. It's going to come back. But we're going to get rid of it for right now. Now, just for a second before we go on, I got to point out something about the microtubules. You notice there are 2 types. There are microtubules that are attached to the kinetochore of the chromatids. Those are called kinetochore microtubules. And then there are microtubules that are not connected to the kinetochores, and those are called non-kinetochore. Another word for non-kinetochore, by the way, and actually I kind of like this word better, is polar. The reason we call these polar microtubules is if you notice they go from one pole to the other. Now, they're not continuous, so this one comes from that pole, and this one comes from that pole, but they're not connected to a kinetochore. So they span the distance in an animal cell anyway from one side where the one centrosome to halfway across, and then the other one from the bottom comes up. We'll talk a little bit later about why these things are different, but I want you to understand that there are 2 different types of kinetochores. Or I'm sorry, microtubules. Prometaphase under a microscope would look something kind of like that. I'm not going to have you, we'll do this in the microscope lab, the mitosis lab. We're going to practice identifying the stages. I'm not going to expect you to know prophase from prometaphase. You can't really tell under the microscope, but you can see right here, this is certainly further along in prophase. The chromosomes have condensed, and you can see, if we could see clearly enough, the nuclear envelope is gone. Don't see the spindle fibers there, though, right? Okay, we're ready to move on to the next stage. Prophase, the chromosomes have condensed. They finished condensing. They finished replicating. Spindle fibers are forming. And in the next stage is metaphase. Meta stands for middle. Meta stands for middle. So during metaphase, the chromosomes, with the help of these spindle fibers, of these components of the cytoskeleton, remember they can act like rails, train rails. Those chromosomes migrate and move until they line up on the middle of the cell. They line up at something we call the metaphase plate or cell equator. There's nothing there. There's no real plate there. It's just the middle of the cell, right? And notice where the spindles are again. We've got some spindles that spindle fibers that are connected to chromatids. So those are the kinetochore ones. And then we have some that are not. We have the polar ones. There's one. There's a polar one right there. There's another polar one right there. So we've got two sets of kinetochores. Here's another picture. Isn't that cool? So you can see the spindle fibers. You can see the chromosomes all lined up in the middle. This is from a rat kangaroo. So these spindle fibers, remember what I told you at the beginning, they're under tension. They're pulling. Those spindle fibers are pulling those chromatids, right? Wanting to separate those chromosomes and pull those apart, but those cohesion proteins are holding them together. Now, when things line up on the metaphase plate, you got to keep in mind that a cell is a three-dimensional object. So they don't line up on a metaphase plate in 2 dimensions. They line up on not a metaphase line, but more of a metaphase plane. I'm calling here a metaphase plate, right? So if you look at that picture there, like the picture up above, sometimes when you look at metaphase, it looks like all the chromosomes, are they all lined up on the middle? Well, they are, but they're on the middle of that of the plate. So they're not all along the same line on that plate, but they're all on that same plane in the middle of the cell. Hey, look up here for a second. Answer me a question. Is this an animal or a plant cell? See if you've been paying attention. How do you know? There you go. I can see the asters, right? There they are. Must be some way I hold this pen that makes it do that. All right. All right, so here we are in metaphase. And again, there's that metaphase plate. Remember, it's imaginary, it doesn't really exist. You can see a really cool electron microscope pictures, the kinetochore microtubules connected to the kinetochores. And of course, there are the non-kinetochore microtubules. Notice they're overlapped, though. That's going to become important. Notice how they're overlapped. And you can see down below where the microtubules are attached to the chromosomes. Really cool kind of pictures. Toboggans with pom-poms. Huh? What looks like a toboggan with pom-poms? That's a good one. Let me go back. This looks like toboggans with... Okay. Okay. And there's metaphase under a scope. Plant cell, obviously, there's the cell wall. Notice once again, it's like they're in the middle, but all of this stuff that doesn't look like it's in the middle it's all along that plane. It's not a straight line. Yeah, I'm not sure I get that with the pom-poms, but if that makes sense to you, Courtney, then by all means. There isn't a phase one, it's a circle. Jordan. So I guess if we interphase is where we usually start. Yeah. So we got interphase, prophase, metaphase. Metaphase is always the easiest one to recognize. The chromosomes are lined up on the middle. All right. Now the next thing that's going to happen. Need to go back. Okay, Emily. No problem. Need to go back a couple slides. Stick with me. Remind you, the next stage is called anaphase, and during anaphase, the cohesion proteins that were holding these together, right there, enzymes digest these proteins. So there's no more cohesiveness. Now remember I told you that the spindle fibers when they're attached, already are under tension. So the only thing that's not allowing those chromatids to pull apart is those cohesion proteins that are holding them together. Now, during anaphase, the stage right after metaphase, an enzyme digests those proteins, get rid of that cohesion, and now those kinetochore microtubules, those spindle fibers, begin to pull those apart. And now those sister chromatids are no longer sister chromatids, but called daughter chromosomes. See them? So that's each of the copies. Cohesion proteins disintegrate and they're pulling apart. And the spindle fibers are what's doing that, right? And if you look at this animal cell and use a little reasoning here, it seems as if what the centrosomes, the asters, and that pair of centrioles, it seems that what that does is it provides like an anchoring point, right? An anchoring point for those spindle fibers, for those microtubules, a place where it's going to pull those chromatids towards. Now, as I said, plant cells don't have centriole pairs. They don't have centrosomes, and they don't have asters. And for many years, we weren't really sure, because plant cells divide. They make spindle fibers, and when we look at the pictures under the microscope or look at the slides, you'll see it looks very much like you would see in an animal cell. But what was it that was providing the anchoring point in the plant cells that they don't have these centrosomes and these asters? Well, you remember one of the big differences between plant cells and animal cells is plant cells have a cell wall. And that cell wall is rigid, relatively speaking anyway. The plasma membrane, which both plant and animal cells have, isn't that rigid. So you couldn't attach those spindle fibers to the membrane, because when they pulled, this is flexible and it would pull it in. Right? It causes it to indent. But if you got a cell wall, we think now what it is the cell wall provides the anchoring point for those spindle fibers. So that's why you don't need them in plant cells. Plant cells use their cell wall. I guess I could try to draw some little chromatids on the end there. Who says I'm not an artist? Okay, so anaphase, there's a middle anaphase and a late anaphase and all that sort of stuff, depending on them pulling apart. What I want you to look at, though, too, is not just the kinetochore microtubules. Look at the non-kinetochore microtubules. Look at the polar ones. This next picture gives you a better idea. In metaphase, those polar microtubules are overlapped. See? They're overlapping one another. As the cell goes into anaphase and as it goes into late anaphase, like you see in the picture, the kinetochore microtubules are pulling the chromosomes apart. The non-kinetochore microtubules, or the polar ones, they're unoverlapping. So they're pushing the cell apart. You see, what's going to happen after all of this is the cell's going to divide too, right? So these polar microtubules are pushing that cell apart. So basically, they're elongating that cell so that later on that cell is going to, oops, sorry, I dropped my mouse. So they're going to elongate that cell so that later on that cell is going to divide, right? See, those microtubules are unoverlapping and they're pushing that cell apart. They're making that cell into an oval. So that's the purpose of the polar microtubules. The kinetochore microtubules separate the chromatids. The polar microtubules are going to push the cell apart. It's amazing to think this goes on in your cells. It's amazing to think it's going on right now as we speak. Here's a good picture of anaphase. Can't see the microtubules real well, but I can tell that's anaphase. The chromosomes are beginning to pull apart. The microtubules are a little bit more visible in animal cells than they are in plant cells. But those microtubules would be connected to the cell wall. And then, of course, there would be the ones that are the polar ones, too. They don't connect to anything. All right? So there's anaphase. Now, at the end of anaphase, what we're going to end up with, let me get rid of my scribbling. What we're going to end up with is we're going to end up with a cell. I'll draw a plant cell to make it easy. With two nuclei. Two sets of chromosomes. One cell with two nuclei. So at this particular point, we finish karyokinesis. All right? The next stage after anaphase is known as telophase. And in telophase, the chromosomes have finished moving to opposite ends, okay? They're going to start unwinding. They're going to decondense. You can't read, you can't use the condensed chromosome any better than you can use your dishes in that box when they're stacked on top of one another. You have to decondense them, okay? The nuclear envelope is going to begin to reform. There it is. But you notice it's still one cell. But see, these stages are dynamic because this is actually telophase, and we notice that cytokinesis is happening. Look at that right there. That's cytokinesis. That's a pinching in, like that picture I showed you at the beginning of the lecture, that's going to eventually separate those two cells in half. Now, the other thing you'll notice if you look in the picture, the spindle fibers are gone, right? The kinetochore, microtubules, the non-kinetochore, the polar microtubules, those are gone. Remember, their job was to move the chromosomes to opposite ends, and now that's all done, so we can get rid of all that. All right? So now we're in telophase, and cytokinesis is starting. Now, cytokinesis, like I said, is going to eventually split that cell in half. I'm going to skip 2 slides and come back to the one I skipped in just a second. I should rearrange the order of these anyway. You know, I'm going to do that right now while I'm looking at my... There we go. So here's our animal cell. Got 2 nuclei. And then there's this contractile ring of microfilaments, right? Microfilaments, again, part of those cytoskeleton that begins like a purse string to cinch in. It forms what we call a cleavage furrow. So as those cells, as those microfilaments get tighter and tighter and tighter, eventually, blip, it pinches those 2 cells in half. Cleavage furrowing. Can't do that in a plant cell, though. Why do you think we can't do that in a plant cell? What is special about a plant cell that cleavage furrowing wouldn't work? Cell wall. Very good. Yep. Got a cell wall. So let's go back to that other slide and see how plant cells do it. What plant cells do, there's the 2 nuclei. There's one. There's one. We're still in one cell. Okay. They form a cell plate. That plate grows until it divides the cell in half. Now, that cell plate is actually not a plate or a line. It's actually a series of holes. I'll come back to that in a minute, too. There it is, right. See it? So these little vesicles, there's a nucleus. There's a nucleus. These little vesicles, like perforations. Like if you took a piece of paper and a straight pin and you held it up and you punched a bunch of holes along the same line, you just kept punching holes. Eventually, you punch enough holes, they would grow together and you could pull the paper apart, right? So these vesicles do the same thing. They start off really small, and then they start growing together, and eventually they grow completely together and we get a whole new cell wall. So animal cells do cytokinesis by cleavage furrowing, blip, pinch in half. Plant cells do cytokinesis by cell plate formation. It looks like a line under our microscopes because of the resolution, but like I said, it's really vesicles. If you look back at that picture, you can see the cell plate again. There it is right there. Identification of a cell plate when you look under a microscope is a good indication that cell is in telophase and cytokinesis. Remember, these stages happen kind of, at the same time, or not the same time, but one's finishing, the other one's beginning. There's the differences, right? Animal cells by cleavage furrowing. There's some animal cells down below. What a neat picture. What a really cool picture, man. These are actually collagen fibers, and they're just about to split that cell in half. God, I love that stuff. It's amazing to me how they can get these. I mean, when I went to biology, when I took biology, this would have been a black and white kind of boring drawing. This is gorgeous. They can do it this way. And then plant cells by the cell plate. Okay. Think you're pretty good at identifying stages of mitosis now that we've gone through all of them? If I showed you some cells, some animal cells, these are animal cells. Oh, these aren't. There they are. There's a bunch of animal cells, very similar to the ones that you see you're going to see under the microscope when we do the lab. What stage is that? Now, if you think, where the heck, this is the cell. It's the same stage that this one's in. This one. The only one. Perfect. Perfect. How about here? Nice. Everybody agreeing with Victoria and Courtney? I don't hear anybody else saying anything. Oh, Courtney, the picture of those two cells dividing? Yeah, kind of. All right, here. Sometimes identifying the stages of mitosis is a process of elimination. So you say, well, it's not metaphase, it's not anaphase, it's definitely not telophase or cytokinesis. If you want to see a telophase or a cytokinesis, down here's one. Right? 2 nuclei. You can still see the chromosomes, so they're still condensed. That means we're still in telophase. They still need to decondense, and then cleavage furrowing has already started. Jordan says pro-metaphase. Could be for this one over here, but remember, I'm not going to ask you because you can't really tell prophase from prometaphase, so the best bet for that, this would be prophase. That's a prophase, that's a prophase, so is that. These are all prophases. We already talked about. There's another one. What's that one there in the upper right? Good. Now, what's that one in the middle? What stage is that in? All right, Seth, explain why you say that's interphase. You're right, it is. Yeah, you can't see them. They're not condensed, right? Yeah, there you go. Perfect. Exactly. And as you'll notice, even if you look at this picture, and when we look at the slides, most of the cells that you see in this picture are going to be in interphase. Most of the cells that you're going to see underneath the slide are in interphase. Remember, cells spend most of their time in interphase. And the way they prepare these things is they take this is whitefish. So they take a tissue layer from a whitefish, and they treat the cells with acid to kill them. And it kills them in whatever stage they're in. And then they put that onto the slide. So since cells spend most of their time in interphase, if you're killing all the cells, you're going to catch most of them in interphase. So that's going to be the most common phase that you see. Right? Very good. Yeah. All right. Last couple slides is going to involve some things, or well, the next slide anyway, is going to involve some things that we talked about before several times in all the chapters known as regulation. Your cells aren't always dividing. Right? Sometimes your cells are doing cellular things, and sometimes your body's going to want to control the cells dividing. For example, if the organism is in a poor nutritional state, it's not going to want to make more cells because that's more mouths to feed, essentially. So there's got to be a way to control the cell cycle. Am I? I'll wait for a second, see if I get caught up. And the cell cycle is controlled by a series of proteins, regulatory proteins. One of those is called kinases, and the other one's called cyclins. And those two things bind together, the cyclins and the kinases, and they bond together to make something called a CDK. It could be. So a cyclin-dependent kinase, and cyclin-dependent kinases are going to end up creating something called an MPF, or a maturation promoting factor, and this is what's going to get the cell to divide. So look at the picture on the right first. There's the cell cycle in the middle. We're coming out of S, so right here. As we come out of the S phase, one of those proteins, the cyclin, begins to accumulate. That's the purple one. So cyclin accumulation increases. Now, if we can jump over to the graph on the left, you can see that's what that purple line is showing you. Cyclin is increasing. As cyclin, the CDK is always there, but the cyclin is only there certain times. So as the cyclin begins to increase and the CDK that's already there, they will bond. They will fit together. Notice it's kind of like an enzyme and a substrate, right? And when the cyclin and the CDK, the cyclin-dependent kinase, bind, they make that maturation promoting factor. It's that maturation promoting factor that allows the cell to go past G2 into the dividing stage. After the cell divides, then the cyclin and the CDK disassociate, the cyclin degrades, and then until it begins to accumulate again when we come back through the cycle. So if you look at the graph on the left, you see that as the cyclin concentration goes up, soon after that, the maturation promoting factor goes up. After the cell divides, both those drop, and then they go up again. cell divides, they drop, they go up again, cell divides. So this is done on a sequential basis. Cyclin accumulation, cell divides, cyclin drops. Cyclin accumulates, cell divides, cyclin drops. That's how we regulate it. So there are a number of checkpoints along the way to make sure that you're ready to do cell division. And one of these is, of course, the cyclin. If the cyclin doesn't accumulate, then the cell's not going to divide. But that's not the only checkpoint. There's actually a number of checkpoints. Well, not number. There's 3 basic checkpoints. We'll start with the one at the top, the G1. So after the cell is dividing, it's going through G1, and this is when it's growing and differentiating and do all that sort of stuff. During that phase, there's a checkpoint. And what that checkpoint does is it checks for the nutritional level of the cell. Like I said earlier, more cells, low nutrition, low growth factors. That's what all that is. Growth factors actually chemicals in the body that let the cells know that they can go ahead and divide. But if the presence and absence of these growth factors is going to decide whether a cell goes through G1 or not. The idea, once again, is can the organism support more cells? Is the nutritional state of the organism in such a way that it can support more cells? Now, some cells don't pass through the G1 checkpoint and enter into a stage we call G0. G0 is kind of a holding stage. The cell during the G0 stage is still active. It's still doing cellular stuff. It's still creating proteins. It's still doing all those things that we talked about in chapter 4 with all the organelles. It's still doing respiration. It's still doing all that stuff, but it's not involved in dividing. Eventually, though, a cell in G0 will get back on the track, go through the G1 checkpoint, and then move its way through the S. All right, so we move through the S. All right, Seth, have fun in chemistry. We move through the S, and we go down to the G2, and there's a G2 checkpoint. Very important at this G2 checkpoint, right? This is, of course, when the cyclin is increasing, but the G2 checkpoint is going to double check and make sure that all the chromosomes have been replicated. You don't want to be missing a chromosome, right? And it's going to check for the integrity of the DNA to make sure there's no mistakes in the molecule. Now, do these mistakes sometimes get through? Yes. Does the G2 checkpoint, is it infallible? No, sometimes mistakes are made. For example, among human beings, we have a condition known as Down's syndrome. Does anybody know the genetic basis behind Down syndrome? Why does someone have Down syndrome? What's the genetic aspect of it? Another word, there you go. Thank you, Brandon. Trisomy 21. And what that means, well, Brandon, you tell me what it means. What's it mean when we say trisomy 21? There you go. 3 pairs of number 21 chromosomes. That's really important. You want to make sure that you want to have the right number of chromosomes, okay? So we're going to check in the G2 point for the replication is complete. Everybody's got the right number of chromosomes and the DNA integrity. If you get through that, you got one final checkpoint, and that's the M checkpoint. The M checkpoint makes sure that each chromatid has a spindle fiber attached to it. All right, so let me go back a little bit, because this is something we're going to talk about when we get into genetics a little bit later. Get a good picture. Let's go. There we are. All right. Let's talk about this chromosome over here. Let's say that for whatever reason, and it does happen, the cell actually makes it through the M checkpoint, and the M checkpoint does not detect that this spindle fiber didn't form or didn't form properly. Bottom line is, when anaphase occurs, since there's no spindle fiber there, that chromatid, or that chromosome now, is not going to move this way. It's going to stay attached, right, to this chromosome, and it's going to be pulled this way. So when these cells divide, if this was the 21st chromosome, this one will have will lack the 21st chromosome, and this one will have an extra 21st chromosome. right? Only you have one maternal and you have one paternal, but if you have an extra chromosome, so that M phase is very important because it makes sure that those chromosomes get split in half and everybody gets a full set of chromosomes. Now, I'll come back to trisomy 21 a little bit later in genetics, and we'll talk again about how that happens. But it's all about the spindle fibers, and that's all the job of that M checkpoint is to make sure that those chromosomes are all attached to their spindle fibers so that they'll pull apart. Again, keep in mind nothing is infallible, so many times cells can pass through these checkpoints even though they do have a problem. Like, for example, here's some cellular problems. Uncontrolled cell growth is cancer. That's what cancer is. We all have the genes for cancer in us. The genes are known as oncogenes. I'm trying to type, but I'm not on the chat box. The oncogenes. Somebody who is a cancer doctor is an oncologist. Right? You have oncogenes in you, right? It depends on whether they get turned on or not. And there's things that turn them on. Exposure to certain chemicals, smoking, a genetic predisposition. But what cancerous cells, what we mean by uncontrolled growth, is normal cells have a couple of characteristics, properties. For example, they have anchorage dependence. Cells require a surface for division to occur. They can't divide out in midair. So in the case of organisms with tissues, a layer of tissue provides the surface for the next layer of cells to form on top in nice layers, okay? And so these cells are going to divide, and they're going to fill in that single layer. And they have what's called density-dependent inhibition. What we mean by that is once they filled in that layer, they're going to stop. Once cells have filled in that one layer, they're not going to divide anymore, they're going to finish. All right. Density-dependent inhibition then means if a gap forms, like if you remove some cells or you get an injury, then the gap is going to be filled in and then it's going to stop. Density-dependent inhibition. So normal mammalian cells look like in the picture as a single layer of cells. Now, what cancerous cells do is they start forming cells on top of cells. In other words, they've been transformed, genetically transformed, and they no longer have density-dependent inhibition, right? So they'll start growing on top. Now, I mentioned before how tissues are formed, cells on top of cells. That's not the same thing. Those are organized layers of tissue. This is lumps of cells that come together. This is a tumor. Yep. Yeah, I think trisomy... 14 is another one that is fatal. Much more fatal. I think it was 14 or 16. Yeah, trisomies can occur in a number of chromosomes. Anytime they don't split apart, they can. 19. Okay. So we get this tumor here, right? What's wrong with a tumor? Well, the problem with a tumor is there's lots of cells in a tumor. And when tumors grow really big, like, for example, if we're looking at breast cancer, 1 in 8 women in the United States will contract breast cancer. It also happens in men. Not as a higher rate, but you have a 0.1%, 1 in 1,000 men. But here's the tumor growing in a woman's breast, right? The tumor gets bigger, and it begins to invade the surrounding tissue. Now, the tumor and the cells in the tumor, they need nutrients. They need oxygen. They got rid of waste products. They begin to hijack all of those nutrients and all of that machinery around them. So they basically begin to kill off or destroy, to kill off the healthy tissue. They actually engage in necrosis. The tissue begins to die. The healthy tissue. The tumor doesn't begin to die. The tumor's having a good time, and it's growing, but it's sucking up these nutrients from all the healthy tissue around it, and it's causing the healthy tissue to be destroyed. So if you go to your doctor, you find a lump in your breast, which you should be checking yourself for that, obviously. One of the things he's going to want to do is a biopsy. He wants to know whether that tumor is benign or malignant. Now, benign tumors are tumors they're cancerous, but benign tumors are tumors that are found there in the breast, let's say. You remove them, and for the most part, you're all right. A malignant tumor is a little bit different. Malignant means bad, right? The reason why malignant tumors are bad is because they undergo something called metastasis. Metastasis means a piece of the tumor, some of these cancerous cells, break off and begin to spread through the blood system, through the lymphatic system, they begin to spread throughout the body. And as they spread throughout the body, they can actually set up shop. They can actually become a tumor in another part of the body, right? So when the doctor does the biopsy, if he finds that the tumor was malignant, then there's that old thing. My mother had breast cancer a number of years ago, and her tumor was malignant. And the rule of thumb among oncologists, kind of, I guess at least it used to be, was if you were cancer-free for 5 years after having that malignant tumor removed, then presumably they got it before it metastasized. Many times when women have breast cancer, they'll have a radical mastectomy, or even sometimes a double radical. Their breasts will be removed. They'll not only take the breasts out, but they take the lymph nodes that are nearby to just try to be sure to make sure if those cells have made it into the lymphatic system that you remove them too. My mother had breast cancer, was cancer-free after 5 years, figured everything was fine. 5 years later, it came back as bone cancer, and my mother died eventually from the complications from bone cancer. So 5 years is a rule of thumb, but it's certainly not set in stone. The idea is those metastasized cancer cells drifting throughout your body can find many years later someplace to set up shop and then begin doing the same thing that they were doing in the breast. And there are certain cancers that when they metastasize, tend to metastasize in certain places. I believe breast cancer, when it metastasizes, very often becomes bone cancer, if I remember correctly. All right, any questions on any of that? All right, I'm going to stop the recording. That's it for the lecture.